Hello and welcome to a new live interview for Cascades Analytics. My name is Alexandre Stepanovic. I'm your host and the founder of Cascades Analytics, a consultancy and media bureau based in New York City, where we're trying to get as close as possible to the action with the development of new psychoplastogens and psychedelics. And I'm really excited to welcome today Sam Banister, who is the Chief Scientific Officer and co-founder of Xylo Bio. Hi, Sam. How are you doing today? Thanks for
Hey.
joining us.
Yeah, hi Alex. Thanks so much.
Yes, so I have a few questions, general questions, and then we can we can dive in the pharmacology of XYL 1001, which is super interesting. But foremost, I wanted to talk about the purpose of Xylo Bio, and so Xylo Bio aims to treat root neural dysfunctions and not just manage symptoms. So that's part of an ongoing shift in psychiatry. So what's hardest about proving that in preclinical work?
Yeah. The whole focus of the company is really to sort of leverage the underlying sort of pharmacology that we're seeing with late stage sort of psychedelic clinical trials, so sort of phase 2 and phase 3 trials with drugs like psilocybin, 5-MeO, DMT, and others. To leverage those sort of mechanisms and our understanding of how those drugs are working, but for drugs with a slightly different profile subjectively. So to achieve some of the same sort of therapeutic outcomes, but without necessarily having a fully hallucinogenic agent. And part of the challenge there is that obviously we have sort of limited preclinical models for psychedelic drug action. We have an incomplete understanding of how psychedelic drugs are working at all. And so there are a number of sort of translational issues moving from preclinical work into the clinic, regardless of how promising sort of either data set might be. So some of these things are unique to sort of psychedelic adjacent drug development, whereas others are just more generally a challenge for psychiatry. We don't exactly have, for example, a good mouse model of depression. So it's very hard to sort of develop antidepressants using any single sort of test or battery of tests that we've used historically. So we have to come up with sort of new approaches here.
What type of issues usually do you run into?
Well, there isn't really a completely concordant set of models or batteries that we can use in mice that represent even a single sort of depressive disorder in a human population.
Mhm.
So we have to rely on sort of composites of um, looking at how a given drug candidate can modulate behavior in a few of these different rodent models, but also looking at other sort of systems levels effects. So looking at various neurocircuitry that might be modulated and circuits that we know are involved in sort of mood disorders more generally. So we have to take a sort of, I guess, a more rounded approach to the way we're thinking about translation than we might in something like oncology, where you might be able to generate a pretty compelling mouse model of a particular cancer type, genetically or otherwise.
Right. And you combine medicinal chemistry and computational design at
Yeah,
Xylo Bio. So, how exactly is computation spinning up discovery for you?
yeah, we've used computation in a number of different ways at
Mhm.
everything from sort of ligand design, ligand protein interaction, which is the more medicinal chemistry end of things, all the way through to some of the analytics that we're using across, electrophysiological techniques like EEG and even scoring animal behavior in some of the video scoring based methods that we're using. So on the medicinal chemistry side, we've used sort of proprietary models of the 5-HT2A receptor and other receptors in that family. So these have been built out, in silico using experimental structure activity relationship data from the libraries. And then we've sort of used that to guide generation of a sort of orthosteric pocket on the 2A receptor as one example, such that we can use that for both high throughput screening of new bits of chemical matter. So these are entirely new chemical scaffolds that look nothing like any known sort of psychedelic or other agent that interacts with 2A. But we've also been able to use those models to sort of rationally engineer key interactions between an engineered molecule and the receptor in a way that we think drives a precise sort of receptor dynamics and signaling process that will lead to therapeutic action, but not necessarily hallucinogenic drug action.
So your screening, was it HTVS?
We have done some of that for sure. So we have one particular family in our pipeline that's sort of partially funded through a government grant in a partnership with another company in Australia, Xylo Therapeutics, and the University of Sydney. And that was really a high throughput virtual screening approach. So can we find new bits of chemical matter based on the 5-HT2A receptor site that we've developed that are very far away from anything you'd identify as an existing sort of 5-HT like scaffold. So in that process, we built out the model, which took some amount of time. We screened about 100 million compounds, which was enough to get a reasonable sort of enrichment of hits. And then we went back and looked at several of those scaffolds that we identified, built out a small library of analogs of each, and experimentally validated that they were in fact 2A agonists. So we've been able to use that as a sort of proof of concept, but also to select at this point some of the leads for that particular series focused on new treatments for addiction.
For addiction, okay. The paradigm for 5-HT2A is shifting a little bit, but we'll come back to that with when you'll be presenting more XYL 1001. So I'd like to continue a little bit on you mentioned using AI differently than other non-hallucinogenic biotech groups. So what's unique about your pipeline?
Yeah, I think we're using a lot of machine learning tools, and obviously, with the current sort of hype bubble that we're in with AI, remains to be seen how speculative it is. But currently, there's still a lot of enthusiasm there. I think almost every company in drug discovery is using AI in some way. So we were joking with an investor recently that it'd be like this investor claiming that all of their portfolio companies are spreadsheet-enabled companies. I think there's an element where people are just throwing AI into pitch decks and trying to leverage the buzzword and the hype just to raise money or for other sort of optics reasons. So the ways in which we've used sort of machine learning approaches are around sort of a clustering and selection of different bits of chemical matter in the high throughput virtual screening, for example, but all the way through to some of our sort of behavioral analyses as well. So we work with Nick Everett at the University of Sydney, and his lab's been using a lot of ML tools for a long time to sort of customize ways of scoring animal behavior. So whether it's sort of opioid withdrawal, using high frame rate cameras and sort of trying to automate a lot of the scoring of those sorts of behaviors through to a sort of platform that we've developed internally around head twitch response, this predictive mouse model of likely hallucinogenic activity. So we've used a lot of sort of ML tools there, really just to sort of speed up and automate a lot of this scoring, which increases the throughput and even some of the sort of sensitivity and accuracy of detections for some of these screening platforms.
And that worked well.
Yeah, it worked extremely well. So we sort of co-developed this with Grant Glatfelter and Mike Bauman at NIDA. So they've been sort of pioneering using high frame rate camera work for scoring head twitch response for quite some time. And we've really just sort of built on that. They published a sort of open source version of this recently that others are free to use. And we've been sort of building on that as well, just to improve our own sort of development pipeline. It's been quite successful.
Head twitch, we know that there are some false positive. MDMA induces head twitch. It's not hallucinatory. So how do you, avoid that false positive?
Yeah, we could spend an hour talking about probably head twitch alone. We've certainly spent a lot of time briefing other groups on exactly what this means. So but I'm guessing most people who are listening would probably be familiar with this, but it's a predictive model that for mice and for rats to some extent, whereby known sort of psychedelic 5-HT2A agonists induce this characteristic, dose-dependent and sort of time-dependent head twitch response that's not typically in the mouse's normal behavioral repertoire, sort of side-to-side or rotational head shaking. So it's definitely in response to the drug. It follows a clear dose-dependent profile, that disappears as the drug wears off. Obviously, it's actually highly predictive as animal models go, with a few caveats. So it's worth noting that really all of the drugs that have been screened in head twitch response really fall into two sort of broad categories. So there's tryptamines, even if you want to include sort of ergolines, like LSD is a special case of tryptamines, and phenethylamines. So the big curiosity is around, once we move beyond those scaffolds, like we're doing with some of our programs, if you move into an entirely new area of chemical space, can you still rely on the head twitch response to be as predictive as it is for other chemical classes that have been well characterized? So that, yeah, there are a lot of false positives. So yeah, one enantiomer, if you followed Bill Fantagrossi's work from a number of years ago, one enantiomer of MDMA, but not the other, and not the racemate seem to induce head twitch. If you go back to Corn and Pickering's early work, where they were in the 60s trying to work out exactly what methods they could use to characterize LSD, they have a whole bunch of false positives that are not psychedelic related at all in that early work as well. So there's certainly a lot of false positives. Luckily, there's very few false negatives. So there's no compound that's been found to be negative in head twitch response that's subsequently been sort of taken by a human and is in fact fully psychedelic.
And
So
that's what you're looking for, yeah.
no, we're hoping to avoid that. Yeah,
Do you want to avoid that? But
yeah,
that's your level of validation for your compound.
yeah. We can take some confidence in the fact that there are false positives. So if we find a positive head twitch, we can look at additional methods to sort of de-risk the compound and convince ourselves that it's not going to be hallucinogenic in people. But there are very few, as I said, no known false negatives. And we found a lot of true negatives, we believe, short of going into the clinic. So it still does have some predictive utility, I believe. It's just really recognizing their limitations. It's definitely not a sort of entirely generalizably predictive model of the likely hallucinogenic activity of all drugs across all chemical classes, including sort of newly developed classes.
Do you use the same mouse across different behavioral experiment for helplessness and head twitch to maybe assess the specific behavior or something?
Yeah, it's a good question. So there's a really nice review on sort of DOI, one of these historical sort of tool compounds that's psychedelic, that looks at the various sort of studies that have used DOI for head twitch in different mouse lines. And I think what's not widely appreciated is that we use C57 black 6 mice for head twitch.
Right.
That's for sure one of the most sort of widely used strains that people are performing head twitch studies in. But historically, people have used other different mouse strains for looking at head twitch, and even strains of rat. And in fact, you get pretty big differences in the head twitch profile, even of a single drug like DOI, across strains and across species. There's been a good bit of really nice work recently looking at sort of sex differences for drugs like psilocybin, and even there's a big difference in head twitch between the sexes, it appears, when you look at pretty decently powered studies. So yeah, I think part of that, just highlights some of the challenges with head twitch is that, we know there are differences in between species. Differences with drugs. DOI gives a much higher sort of total head twitch count than something like psilocybin, and the reasons for that are not entirely well understood. So I think it's pretty critical that we try to use head twitch with as many consistent parameters as possible when we're doing sort of head-to-head comparisons. So that's been the approach we've taken. These experiments are run in the same lab with the same mouse strain. We often run head-to-head with various positive controls, so we can get a really good handle on exactly what sort of profile we're getting for a new drug.
And how do you see your neuroplastogens fitting into the mental health care long term? Faster relief, longer remissions, something else?
Yeah, we're in a position now where there's certainly a lot of interesting emerging antidepressant technologies. If you look at sort of even non-drug interventions, so transcranial magnetic stimulation and some of these other emerging modalities are certainly looking promising. I don't think there'll ever be a one-size-fits-all sort of solution for depression, broadly defined. I mean, depression is already very likely a number of different subclassifiable disorders. We sort of know that even with the limitations of the existing DSM framework. So I don't think there's ever going to be a single antidepressant solution that works for anyone. What we're aiming for here, psychedelics are obviously, if you look at Compass's sort of phase 3 data, Gilgamesh's phase 2 data, that there's clearly going to be a role for fully psychedelic agents that are, ideally short-acting, administered under medical supervision in a clinic for people with major depressive disorder or even refractory kind of treatment-resistant depression. But there are also people that are contraindicated there. So if you have a family history of sort of acute psychotic episodes, schizophrenia, maybe you just don't want to have a psychedelic experience, and the person I think about is, my own mum being treated for an anxiety disorder or something. She doesn't drink. She's never tried a psychoactive substance. She's probably not going to want to go into a clinic and have a fully psychedelic experience. Obviously, SSRIs, SNRIs, these other sort of atypical antidepressants that are being used, tricyclics and things that have a long history of use, none of those are perfect either. So we know that people who are treated on SSRIs, for example, have this sort of latency of onset. So you're looking at several weeks in most cases before patients will start to feel any relief from those drugs. They certainly don't affect everyone equally. So, it's estimated that maybe sort of about 1 in 7 people maybe get adequate relief from SSRI treatments. And so the paradigm we're stuck in is that people are essentially just going through trial and error in psychiatry with one drug after another until they kind of find a treatment that works. And there's obviously a sort of weaning off period as drug substitutions occur. So you're left with this position where ultimately SSRIs are some of the most overprescribed drugs on the planet. Probably very few, a minority of patients in a literal sense is getting effective relief. They take a long time to work, and there are a whole bunch of different side effects with these things. So people often report sort of emotional flattening. So they're no longer sort of truly anhedonic, but they're not getting much pleasure out of things either. It's not a complete anhedonic state. There's a lot of sexual dysfunction and sort of increasingly this awareness that in some cases sexual dysfunction persists even after the drug is stopped, which is pretty interesting and sort of understudied. So what we're looking for here is something that works sort of as we think psychedelics are working in the clinic, which is that people report relief within 24 hours of their dosing session. They often have that single treatment session or 1 to 3 treatment sessions, several sessions in succession, and then have relief for weeks or months out in some of these clinical follow-ups. So we'd like to be able to have a drug that is capable of doing something very similar, but without the fully hallucinogenic profile of these substances. So this might be a single treatment, it might be an episodic treatment, but it's delivered by the patient under the sort of supervision of a doctor in terms of prescription, but at home, in the comfort of their own home. They don't have to go to a clinic. They can just, take the medication as directed, like any other tablet, and have relief, without all of the side effects that currently sort of afflict SSRIs.
I'm wondering if ketamine is having a fantastic effect. Apparently, I mean not apparently, but is leading therapeutics in terms of antidepressant treatment, but it's a different route than it's different mechanism of action than serotonin. So I'm wondering if the psychoplastogens who actually interfere with serotonin signaling will be blunted because they'll have to activate within the patients a serotonin signaling that's already being damped by years of SSRIs use. I don't think we know that yet, but what do you think?
Yeah, it's a really good question, actually. And obviously, most of these changes are probably not permanent. We know that the brain is highly sort of homeostatic. So if you stop taking a drug over time, we know this from various ADHD medications where imaging studies have been done, certainly for some antidepressants. The brain will eventually renormalize to a state without the drug if you stop taking any given drug. That sort of tolerance or dependence can disappear. Sort of tapering off drugs is why this is done in a slow way. With psychedelics, some studies have actually been done where patients are still using SSRIs whilst being enrolled in psychedelic clinical trials. And what's interesting there is there's actually not a really dramatic difference in terms of the sort of subjective profile of the drugs or the therapeutic outcome. So that sort of gives us some early evidence that, in terms of safety and definitely in terms of efficacy, that there may not actually be such big differences between patients who are on SSRIs and those who are not when they're using psychedelic drugs, and then potentially even other sort of 5-HT targeting neuroplastogens. So it might be possible to have, this is all speculation, there's no clinical evidence to support this yet, but it might be possible to have sort of enrollment on a new neuroplastogen drug once medically approved, as you taper off another medication, or to sort of wean off one and start taking the other at increasing doses. So there's certainly early clinical evidence there that it's not entirely incompatible in terms of an SSRI treated patient and psychedelic therapeutics.
So let's dive into XYL 1001. So it is a non-hallucinogenic 5-HT2A selective neuroplastogen. Mechanistically, what separates it from classic psychedelics?
Yeah, so we've taken a really, as you can sort of tell by the way we've spent a lot of time building out proprietary models of some of these receptors, what we wanted to do with XYL 1001 is take this really target-based approach to development of a clinical candidate for the treatment of depression. So most psychedelics, or all psychedelics as far as I'm aware, will hit the 5-HT2 family, so 5-HT2A, 2B, and 2C at the very least. Often things like psilocin, the active metabolite of psilocybin, also hit 5-HT1A, a number of the other 5-HT subtypes. And for drugs like LSD, there's also activity at sort of alpha adrenoceptors, dopaminergic receptors. So these agents are pretty characteristically defined as being polypharmacological in nature. We know that the 5-HT2A receptor is the critical receptor for the psychedelic drug action. And this has been demonstrated even in humans, clinically with antagonist experiments and things. So you can essentially prevent a psychedelic experience with LSD by pre-treatment with a selective 5-HT2A antagonist. You can also terminate an LSD experience with the post-administration of that same sort of antagonist. And this has been shown, I believe, by Matthias Liechti's lab in Switzerland and a number of other labs as well. So we know this is the key receptor. Our approach here was, if this receptor appears to be key for the psychedelic drug action, if it appears to be central for a lot of the neuroplastic effects that we're seeing with these substances, and then obviously, there's a mixture of sort of preclinical data there, but some of the more compelling data sets certainly suggest that the neuroplastic effects in the cortex that the psychedelics are inducing in rodents are highly 5-HT2A dependent. So one of our advisors, Alex Kwon, has shown this very recently in a nice Nature paper, where if you generate 5-HT2A knockout animals, where the 2A receptor is not expressed in these key pyramidal tract neurons, you actually lose a lot of the neuroplastic effects of drugs like psilocybin. You also lose a lot of the sort of translational antidepressant-like effects in some of these key behavioral models. And this can be shown in the same animal using in vivo microscopy techniques, which is pretty neat.
It's a full knockout.
Yeah, it's a conditional full knockout. So the approach we've taken is let's aim to develop. So some drugs are more selective for the 5-HT2A, B, and C receptors over the others, but nothing's ever really been developed that's truly selective for 5-HT2A. The closest examples would be some of these NBOM, NBOH type compounds, 25CN, NBOH, or 25I, NBOM as an example. They're more selective for 2A. They're fully hallucinogenic, but no one's ever developed a non-hallucinogenic 2A selective agonist. So that was really the aim of the program: is let's develop this agent, rationally engineer it to be likely safe and tolerable, to be suitable for human dosing, and build out a preclinical data package to support the ultimate clinical development of this agent. So XYL 1001, it's about equipotent with other psychedelics. So it's about 20 nanomolar EC50 at 2A, which is pretty similar to psilocin. No appreciable activity at any other 5-HT receptor. No appreciable activity at about 90 other receptors that we've looked at this point, both CNS and other. So it does appear to be truly selective. It's non-hallucinogenic in our wild-type mouse head twitch response up to about 43 milligrams per kilogram, which is as high as we've taken it. For reference, psilocybin's definitely showing effects at about 0.5 milligrams per kilo in that sort of paradigm. So appears to be non-hallucinogenic. It induces neuroplasticity in a 5-HT2A dependent way, and we've shown this using a neural stem cell line, differentiated neural stem cell line, where we can actually silence the expression of 2A. So once we do that, we don't see any of the sort of increased dendrite outgrowth that's characteristic of a lot of these psychedelic and other 2A agonists. And behaviorally, we're getting a profile that's very similar to some psychedelics. So following a single dose of this drug in a number of mouse and rat models that are commonly used in sort of translational psychiatry, we see effects within 24 hours, like you do with psychedelics. And often these effects persist up to sort of 7 or 8 days, which is typically how far we've looked in any of these models. So we have some very recent data that looking at EEG. So we're really interested in how do we translate sensible dosing and activity into the clinic with a drug that doesn't have some of those clear pharmacodynamic markers that a psychedelic has, positive head twitch and other things. So we've been using a lot of EEG. We have some really nice EEG data to show that we do get increases in these key spectral bands, frequency bands that psychedelics also tend to increase, but we're not getting other sort of markers of psychedelic action like suppression of alpha activity, which seems to be pretty common to psychedelics, but also to things like ketamine, Spravato. So we think we have a very differentiated profile here. It does appear to be a truly 2A selective, non-hallucinogenic compound. And we're in the process now of doing all of the sort of IND enabling steps that will allow us to take it into a phase 1 trial in currently scheduled for Q2 of next year. So we're pretty excited about that.
Wow, it's yeah, fascinating. Congratulations. It sounds almost miraculous. Tell me the EEG. Can you tell by reading the EEG if there's ego dissolution? I mean, desynchronization of cortical networks, hence possible ego dissolution?
Yeah, we weren't looking at EEG. We're looking at several different biomarkers. Some of which I can speak about EEG because others are looking at it. We're also looking at some other pretty interesting 2A dependent peripherally released biomarkers that might be of interest, where there's some clinical support for these as well. But yeah, I can say less about those. But for EEG, we weren't really looking for a translational marker of necessarily therapeutic activity. What we were really looking for was some understanding of the sort of dose dependent profile of the drug, and to see if we could sort of benchmark that to some of the frequency bands for known psychedelics. So, this idea of developing a non-hallucinogenic drug, if you want to develop another psychedelic drug, which is, sort of what Gilgamesh has done with bretisilocin, you have a lot of benchmarks that you can point to. So does it produce a head twitch that's somewhat like other psychedelic drugs? Does it produce EEG signals that are somewhat like other psychedelic drugs? You have a lot of different points of comparison.
Mhm.
If you're looking for a compound that's non-hallucinogenic but does engage the 2A receptor, in vivo, that's a little bit more challenging. So the two approaches we've taken, we have a really elegant set of experiments where we've actually used a known and consistent dose of DOI that induces a reliable amount of head twitch over 30 minutes. And we can show that if we treat with our compound, which has really excellent PK in terms of getting into the brain, is a potent 2A agonist, we can sort of dose dependently suppress the effects of DOI in terms of that head twitch response. So it's a pretty clear indication that even though the compound's an agonist and activating the receptor, we can actually sort of displace through competition the activity of a known psychedelic. So that was pretty good confirmation that we are indeed hitting the target in vivo. There's other things we can do there that we're sort of considering if needed, things like PET occupancy to show that you have a engagement with the target and quantify that. But EEG is really nice because if we can show a difference from saline in a way that's dose dependent, we can show engagement and start to think about exposure of the drug in terms of the effects that we're getting. But the other thing we can do is to start to compare to both non-hallucinogenic polypharmacological 2A agonists, things like 6-fluoro-DET that's been used, or Ariadne, some of these other compounds. And we can also compare to things like psilocybin or DOI or LSD, other known psychedelic drugs. And so what's interesting there is, as I said, we get sort of the same directional changes and similar sort of magnitude in changes in some of these frequency bands that we know psychedelics also modulate. But in other bands that are, they're not strictly correlated with the subjective human experience of these drugs, but there's sort of a common understanding that suppression of alpha band may be associated with sort of dissociative or hallucinogenic states, at least for psychedelic drugs themselves. And so we're not seeing that with our compounds, which is pretty nice. We wouldn't expect the drug necessarily to have an EEG signature that's kind of identical to psilocybin. That wouldn't necessarily make a lot of sense if it's not
Right,
hallucinogenic.
right. That's great. So now the paradigm in terms of how to what is responsible for hallucinations in at the 5-HT2A level. Previously, until 2022, 2023, the paradigm was that it was the GQ pathway that was favored over the beta arrestin pathway. But then studies started to show that you could have high activation of the beta arrestin and still have hallucinations, like with LSD, for example, or the other way around, having a strong G2 activation and no hallucinations. So that paradigm seems to be uh, being revisited, and now it looks like more the localization of the 5-HT2A is important rather than dendritic 5-HT2A, but maybe more somatic 5-HT2A or 5-HT2A receptors based on localized on interneurons could be the one uh, that will still induce neuroplasticity but not trigger hallucination, and the dendritic, the apical receptors would be the one
Mm.
responsible for hallucinations. So that's
Yeah.
one theory. Another one is receptor occupancy that you mentioned. And so, how does XYL 1001 fit in that new paradigm shift?
Yeah, there's so when we think about the sort of models that we've had for drug action historically, we had old models of sort of hand and glove. This idea that things have a binary state, you can turn a receptor on or off, and then with allosteric modulators, we sort of introduced this idea of like a dimmer switch almost for activity at a given receptor. So
Mhm.
I think our models for drug action are actually evolving in a way that's more representative of the complexity that we understand of something. With as many different components, interrelated components as the brain.
Mhm.
So I think it's more appropriate to think of, we think about the 5-HT2A receptor, but it's expressed, postsynaptically in some places,
Mhm.
presynaptically in some places. In some cases, there's a really nice recent paper, I believe from Ahmed's group, where presynaptic 2A receptors may be relevant, but where there's projections for certain neural pathways into other regions of the brain.
Mhm.
So this idea that we're hitting this one sort of family or population of 2A receptors in a way that does a single thing, I think is pretty simplistic.
Mhm.
We know that there's a balance between 2A and 2C for different sort of excitability components with signaling. So yeah, I guess part of the challenge and part of my frustration is whenever some really interesting science is done to show that something appears to be a sort of key driver for likely hallucinogenic or non-hallucinogenic effects. It's likely true for the cases for which that has been demonstrated, but it's probably not fully generalizable. So this is a little bit like head twitch. If you
Mhm.
make another tryptamine with a certain pharmacological profile that's similar to other tryptamines, and you put it in head twitch and get a nice positive readout there, it's probably a psychedelic in people. But if you make a naphthalene or something else, as we've done in other parts of our library, a completely new scaffold that has a slightly different profile, new chemical class, and it gives you a positive head twitch readout. What does that really mean? Until there's a human experiment, we can't really say. So I think this idea that GQ signaling efficacy is part of the story for likely hallucinogenic activity is probably true. There's some really nice data from Sheng Wang's lab in China that was at a Gordon Research Conference recently showing a heavily beta-arrestin biased compound, almost no GQ signaling efficacy. It didn't appear to be head twitch responsive, and they've also done some studies in a macaque monkey model to show that it is very different from LSD.
Mhm.
So, at least for that case, this is a completely new scaffold as well. It doesn't look like any known psychedelics. In that case, beta-arrestin bias clearly is important for its likely hallucinogenic activity. But then there's other papers. Compass has started publishing some of their preclinical data. So there's a pretty nice paper from Aurelia Ippolito showing that when they've looked at a number of different compounds preclinically, there doesn't seem to be any correlation between bias and head twitch response really at all. And the sort of summary of that paper is that the final line is that they don't believe bias is actually super important for head twitch response. And I think it's sort of a bit of a intellectual trap to fall into to assume that there might be just one single answer that applies to every single situation in which we're trying to study hallucinogenic activity.
Mhm.
So we're looking at the bias now for our compound because we have hypotheses about which pathways might be relevant in terms of receptor dynamics and the sort of downstream signaling that results from that.
Mhm.
But it does have some GQ signaling efficacy by several different pathways. We've confirmed
Mhm.
that in several different endpoints.
Mhm.
It's sort of right around, but a little bit above that threshold that was proposed in the really nice paper by Jason Wallach and John McCorvey, where that GQ signaling efficacy seems to be critical. So we given that it's a new scaffold with a new profile, and it's very selective for 2A, it might not actually be either of these stories. It might be that it's a different sort of aspect of the bias that's important,
Mhm.
or the particular population of 2A receptors that are being activated in a particular way by this sort of functionally selective chemical scaffold.
Yeah, it would be nice to see where in the brain does those compounds bind at the receptor level. Unfortunately, there's no good antibody for 5-HT2A, but there's other ways to look at that.
Yeah, so someone could raise one. There's definitely ways to do that. You can also use PET imaging, but that will show you interactions in a living system, but it won't necessarily tell you about sort of differences in activation within different sort of populations of 2A receptors.
You could do, yeah, you could do PET using LSD washed by
Yeah.
Xyl 1001.
Yeah, so there's sort of good structural modalities that we can look at, and then there's things that aren't as sort of aren't as discrete in terms of the resolution for structure, but are maybe more useful functionally. So fMRI and EEG, some of these other tools that we're using.
How do your compounds, especially XYL 1001, differ from others like Gilgamesh's and or Helix's?
Yeah, one of the I think it was Jackie from Solera had a nice post on LinkedIn the other day saying, drug development's a funny business because if you're aiming to treat really serious life-threatening illnesses in the case of serious depression, it's hard to not want to rally around your so-called competitors because more drugs that are available that are safe and effective means better treatment outcomes for patients generally. So. With full credit to the Delix team and Gilgamesh team and a lot of respect for their science, I think one of the nice things about this field is that everyone's taking a slightly different approach to the same problem. So, successes are good generally in terms of sort of advancing the science and developing new medicines for patients. But a failure from any one company or approach also doesn't mean that the whole field is likely to struggle. So Delix, is fairly different, and they've really focused on measures of sort of structural plasticity for their drugs. So it's a more phenotypic based sort of approach to discovery, which is totally valid. That's how many psychiatric medicines were discovered historically. And Gilgamesh, they do have a neuroplastogen program. It's hard to tell exactly what the focus of that might be. It might be based on sort of Ariadne and some of the work that the team was doing in Dalibor Samesh's lab at Columbia. Or it could be something totally different. But for their psychedelic drug program and their sort of reduced dissociation ketamine program, these are essentially analog programs. So you start out with a drug that does something and works pretty well, make minor tweaks to it to either improve PK, or other aspects of its sort of activity or subjective profile, and hope that the efficacy of the proven drugs holds out. So one sort of a phenotypic approach, one sort of an analog-based approach. I'd say ours is different in that we've really taken this more sort of target-based approach. So as far as I know, we're the only group that's really focused on truly building a functionally selective 2A activator that is non-hallucinogenic and demonstrates the same sort of profile that many of these psychedelic drugs are showing in the preclinical models.
Mm-hmm. And then one last question. What's next on Xylo Bio's research horizon?
Yeah, that's a great question. So, our website has some of the sort of pipeline displayed there, but obviously psychiatry is a huge unmet need. We occasionally get sort of feedback that the depression is a challenging area, which is true. But one of the things I think is most exciting about sort of this rediscovery or renewed focus on the 5-HT system is that psychedelics themselves are showing activity in some pretty interesting areas, and often these are areas that haven't been looked at since prohibition. So there's a really nice study very recently by Ellen Bradley and Josh Woolley, for example, at UCSF, looking at the effects of psilocybin on sort of mood impairment and other aspects of mood, depressive-like symptoms in Parkinson's disease patients. It's an open label small study, and that they're doing a larger study now with psilocybin. But one of the interesting findings from that study was that, beyond the sort of improvements to mood, which you might expect given what psychedelics are doing in MDD and TRD, they actually got improvements in sort of various cognitive symptoms and also in motor symptoms as well. So, it's a small study that they're still sort of increasing the patient size there and looking in a larger, more representative patient population. But I think, taken in parallel with some of the work that the Gilgamesh team did on Ariadne and in a different rodent model of Parkinson's disease, the use of psychedelics and these sort of neuroplastic mechanisms in areas of neurodegeneration, potentially Alzheimer's disease, frontotemporal dementia, like Solera is looking at, these other companies. I think that's a super exciting area. I'm also really excited about migraine disorders. So you have Emmanuel Schindler at Yale doing some really excellent work there, and that's sort of back translation from a number of different migraine and cluster headache sufferers reporting on their own success anecdotally with different psychedelics. And the other study that's pretty exciting, I believe, is in sort of areas of chronic pain. So every now and again, there's one just earlier this week, looking at various chronic pain models and the effects of psychedelics in those. So there's at least one really nice clinical trial going on at UCSD, looking at phantom limb pain. with psilocybin, and that's again based on sort of anecdotal reports of patients with various chronic pain conditions finding efficacy with these drugs. So we've sort of built our pipeline around areas in psychiatry generally. I mentioned we have that substance use sort of addiction program that's at the discovery stage, but we're increasingly interested in these other areas outside of psychiatry, so moving into neurology. So we do actually have a program that's focused on chronic pain, and we've demonstrated, for example, that for both a selective and a non-selective 2A agonist, we've got a couple of different leads there in our pipeline. We do get really good effects in a number of different sort of persistent pain models in rats. And we're currently building out that sort of data set with a view to raising some funding around that, seeking sort of non-dilutive funding opportunities with NIH institutes, to really sort of advance that novel sort of approach to chronic pain treatment. So again, non-hallucinogenic, sort of psychedelic inspired mechanisms, but in a way that's much more effective than opioids or gabapentin or other compounds for chronic pain, and can be taken at home without the need for a clinical supervision.
Mm-hmm. I see also that you have some 5-HT6 agonism. So that is that related to? I know that 5-HT6 is related to cognition and memory. Was that intentional during the screening, or is that something you found afterwards?
Oh, we do indeed have some in various parts of the library. Do have activity at 5-HT4, 5-HT6, 5-HT7, some of these other targets. We haven't really. We're not actively developing for that. We haven't sort of disclosed a lot of that. I think there might be confusion with Solera's program, where they're very interested in 5-HT2A and 5-HT6 for the treatment of dementia for various rational neurocircuitry-based reasons. So we're not actively focused on that, but I know Xylo is very interested in that sort of combination mechanism.
Their purpose is actually interesting, is to develop non-hallucinogenic to deliver to patients with dementia. So that makes sense here as well. So that's very interesting.
Yeah, 5-HT6 has been a target for Alzheimer's disease historically, with some challenges in the clinic, but that doesn't mean, in combination with 2A and 6, it's quite a different approach. And I think it's a super interesting direction that they're taking their approach to development here.
Well, Sam, thank you so much for your time. That was fascinating. All the best with Xylo 1001 and the other compounds. It's truly promising and super interesting. Thank you so much.
Yeah, really great to speak with you, Alex. Thanks for hosting me.
Sure, and I'll see you next time, maybe in New Orleans. I don't know if you go to the convention.
Yeah,
in February, I think it is.
That's right. Yeah, we'll be attending ACNP in January, and we'll be attending ISRP and the other drug development meeting that John Westliff hosts around that meeting as well. So I'll see you at one of those events for sure.
Okay, thank you.
Alrighty, thank you so much.
Bye. Thank you, everybody, for tuning in. And if you have any questions, comments, or if you want the transcript of that interview with Sam Banister, feel free to reach out directly to me. And I'll see you next time, next Tuesday, actually, for an interview with Chris Witowski of Xylo. Until then, be well. Bye.